This time we're going to cover the third and final stage of gene expression, which is translation. So translation is going to take place within the ribosome. Remember, in transcription, where we built our mRNA molecule, then process it via splicing to make the mature mRNA transcript, that's going to leave the nucleus, travel through the cytoplasm, and arrive at the ribosome. So the tRNAs, they're also going to enter into the ribosome, and they're going to start to translate that message into a protein. Now they'll do that 3 bases on a codon at a time. So the anticodon binding to the codon at one end and bringing in the corresponding amino acid at the other. Okay. So using that base pairing rules, we're going to translate our genetic code, sequence of bases, into a sequence of amino acids. Okay. We'll start with a start codon and end with a stop codon. And eventually, when we've got all of our amino acids, the tRNAs can leave the ribosome and we've got our polypeptide or basis of a protein. Okay. So to start translation, we have to start with a start codon. If you look at your codon charts, you'll see that's AUG, which corresponds to the methionine amino acid. So every protein will start with the same amino acids. Okay? That tells the tRNAs to start reading 3 bases at a time, start bringing in all of the amino acids until eventually one of these 3 stop codons is reached. And that's instruction to the tRNA. No more amino acids need to be added now. We can stop growing our polypeptide. Okay? So the tRNAs stop binding to the mRNA, stop bringing amino acids, and exit the ribosome. So the important features of the tRNA include the anticodon. So the anticodon will be complementary, the opposite to the codon of the mRNA. So G with C and A with U in RNA. So that's how we can go from a sequence of bases to a sequence of amino acids. So bind to the mRNA codon to anticodon. At the opposite end is the amino acid attachment site. So as the tRNA binds to the mRNA, it drops off the corresponding amino acid into the ribosome. So as it does, it drops off its amino acid, exits the ribosome. The next tRNA comes in with the codon and anticodon, leaving the next amino acid. That tRNA exits, goes, picks up more amino acids, and that will continue until the stop codon is reached. So the amino acids that were added by the tRNA, the transfer RNA, they themselves get joined together via peptide bonds. So hydrogen bonds between the bases, but peptide bonds between the amino acids. So as these amino acids get joined together, that's what builds up the polypeptide chain. Once we've got all the amino acids necessary, the tRNAs will exit the ribosome. The polypeptide can exit later, and that's what we'll cover in a subsequent video. What happens to the polypeptide, how it gets into its final protein structure. Okay. So as always, let's try some past paper questions. So the diagram shows 3 stages of gene expression. Which row at the table identifies these 3 stages? Pause the video, hit play when you're ready to reveal the answer. So the correct answer is D. So stage X, we can see we're going from a DNA molecule to a single-stranded mRNA molecule, so that must be transcription. Note how that's happening within the nucleus. Stage Y, so that mRNA transcript is being shortened. still within the nucleus, so that must be the RNA splicing process, leaving stage Z, which is happening within the ribosome, to be translation, where we're going to actually build our polypeptides. Let's try another. So the following sequence shows the order of bases in a segment of M messenger RNA, and you're asked how many different amino acid molecules are coded for by this segment of mRNA. And remember, if you do need a codon chart, one will be provided to you. So do make sure you can use both the table and circle versions of these charts. So pause the video now, come up with your answer. So let's solve this one together. Remember, each mRNA transcript will be read 3 bases or a codon at a time. So a good place to start is just to separate the codons, make it much easier to decipher. So this shows us we've got 1, 2, 3, 4, 5, 6, 7, 8 different codons. Remember, the question is how many different amino acids are present. So if you go to the codon chart, you'll see some are there more than once. Remember, there's redundancy within the codon charts within our genetic code. So different sequences could code for the same amino acids. So you've always got to double check. It's not necessarily the number of codons. You've got to go to the codon chart and see how many different amino acids are coded for. So there's 8 codons, but 6 unique, 6 different amino acids coded for by this particular sequence. Again, hope that's helped make translation make a little bit more sense, and any questions you can always leave below.